Hi, everyone. My name is Alan. I am a PhD student in the Lockheed and Wong Labs in the Department of Biology at Queen's University, and today I'll be presenting my master's thesis research on overwintering water column distribution of the toxic Cyanobacterium, Microcystis aeruginosa. So toxic cyanobacterial harmful algal blooms damage aquatic ecosystems, reducing water quality and releasing deadly cyanotoxins. Blooms are of increasing prevalence, and remediation efforts have been of limited effectiveness, and they've been causing billions of dollars of damages across the world. Microcystis aeruginosa is a common bloom-forming cyanobacterium that can control its buoyancy, and its life cycle consists of a benthic overwintering resting phase, a pelagic growth phase, and a colonial blooming phase. Sources of Inoculum have traditionally thought to be in the lake bottom and sediment from post-bloom colonies that settle in the water column in the autumn. However, the literature indicates that cyanobacterium in general have very high tolerances for freezing and strong low temperature viability. Furthermore, spring growth is thought to be driven by light and wind mixing, and this all supports the possibility of near surface wintering. The water column distribution of overwintering Microcystis and the factors that influence shifts in toxicity remain very poorly understood. And despite this assumption of sedimentary overwintering cyanobacteria, the literature on its actually exploring their distribution overwintering is very sparse. However, advances in quantitative PCR and environmental DNA allows us to explore these questions for the first time. So to explore these questions, we sampled water during the winter between February and March in 2019. So we did this in Dog Lake in South Frontenac County, roughly 30 minutes north of Kingston, Ontario. So this is a water body on the Rideau Canal with a lot of recreational activity and experiences annual blooms. Over the course of 6 sampling days in those 2 months, we collected water samples from the near surface, near bottom, and the ice cover. Our samples consisted of around 300 milliliters of water, and we filtered and extracted them for DNA with 1.2 micron polycarbonate tracked edge filters and a chloroform ethanol method of DNA extraction. We ran qPCR on the samples for the Microcystis aeruginosa 16S gene and the MCYE gene. So not all cyanobacteria are toxic. Even within the same species, there are toxic strains and non-toxic strains. So by looking at the 16S gene, a common barcoding gene, we can examine the overall population, and the MCYE gene, which is a gene in the microcystin production pathway, we can look at the toxic population. We then analyze the gene concentrations with ANOVA's, a general linear model, and linear regressions. So these were our results. In figure 1, you can see overall Microcystis concentration, which is around 10 to the 8 gene copies per liter near the bottom, 10 to the 6 near the surface, and 10 to the 5 in ice cover. Near the bottom, this was around 2 orders of magnitude below the minimum threshold for an algal bloom. And all 3 sources had quite a lot of viable Microcystis, indicating that they were all potential overwintering inoculum. There were significant differences between the 3 sources, but it was very surprising that ice cover had so much, and it was quite close to the surface concentration at times. In figure 2, we can see correlations between Microcystis concentration and the water body depth. So in the near surface population, as the water column deepened, as the lake bed deepened, the Microcystis concentration actually increased, which is very surprising. And as the lake got deeper, the near bottom Microcystis population concentration decreased. So we can see this trend again in figure 3, which has a y-axis of ratio of near surface to total Microcystis along an x-axis of lake depth. So in the shallow areas of the water body, the Microcystis population was very near bottom dominated, and a very small percentage was near surface. However, past around the 16 meter mark, we see a shift to near surface Microcystis, which is a very novel finding. In figure 4, we plotted the ratio of toxic to total Microcystis, looking at trends in toxicity, but we did not see a significant trend near the surface. However, in the near bottom, toxicity decreased with depth, showing that as the lake got deeper, the overwintering population near the bottom of the lake became less toxic, which is again a very novel finding. So overall, we quantified the relative concentrations of toxic and overall microcystis in the near surface and near bottom of Dog Lake at sites with different depths. Our methods were looking towards intact microcystis, so viable cells, because our pore sizes were too large to capture free-floating DNA, and we only captured whole cells. This indicated that there was a viable inoculum in all three sources, and that the ice cover should be more strongly considered, as well as the near surface, as overwintering sources of inoculum. Overall, the microcystis concentration was higher in shallower lake bed areas, which is very similar to the literature, which is likely due to greater recruitment success in the spring. And there was a shift to near surface microcystis dominance in deeper lake bed areas, indicating that pelagic platonic populations may be a more significant source of inoculum than previously thought, especially in deeper areas of the lake, as well as deeper lakes. Water bodies with smaller littoral areas and high sedimentation rates, such as dam reservoirs, might have a lot of planktonic Cyanobacterium as inoculum for summer blooms. Therefore, for those water bodies, we may wish to consider the pelagic area for early season remediation attempts. Thank you for watching, and thank you to my supervisors, Dr. Yu Sheng Wang and Daniel Lefebvre, and thank you to my lab.